Hey guys, in this video we're going to discuss about how to perform laboratory test for testing the presence of starch in leaf. Okay, in previous video we have discussed about photosynthesis, and there we have seen that in the process of photosynthesis, glucose, which is a carbohydrate or an organic material, is being synthesized by all green plants, okay, in the process of photosynthesis. Now this entire glucose is not utilized at the same time by plant. So excess of glucose in plants is actually stored in the form of starch in leaves. Fine. Now how to test the presence of starch in leaf. Fine. So in this video we will discuss about how to test the presence of starch in leaf. Now in order to test the presence of starch in leaf, the materials which you require is, first you require a beaker, okay, then you require a stand on which you will keep beaker. You require green leaf, the leaf in which you're testing the presence of starch. You require the source of heat. Okay, now the source of heat could be a spirit burner. Okay, it could be a Bunsen burner. Okay, it could be a normal electronic heater also. Just we require a source of heat so that we can boil this water, and further on if we can boil alcohol. Okay, so the first step is you have to take a fresh leaf from plant and boil it in water for 30 seconds to 1 minute. Okay, so first what you have to do, you have to take a fresh green leaf from the plant, any plant in which you are testing the presence of starch. Okay, because every plant performs photosynthesis, so every plant will have starch in it. Okay, every leaf of a plant will have starch in it. So pluck one leaf from a green plant and boil it in water for 30 seconds to 1 minute. Now you should understand the reason why we are boiling leaf in the water. Okay, because these questions are asked in ATP papers sometimes. Okay, in alternative to practicals paper sometimes. Okay, so we are boiling leaf in water in order to stop any chemical reactions in the cell and break the cell membrane. Fine. Actually, in Unit 4 biomolecules, you have studied that we perform starch test by using a reagent called iodine. Okay. Now here also we are going to use iodine itself. Now, if you will take a leaf, if you will directly pluck leaf from the plant and add iodine drops over it, what will happen? The iodine solution will not be able to enter inside the leaf cells. Okay, you are aware that where starch is present inside the leaf cells. Okay, in palisade mesophyll cells or in spongy mesophyll cells. Inside these mesophyll cells you have an organelle called chloroplast. Okay, so inside chloroplast starch grains are present, isn't it? So this iodine solution has to reach to the chloroplast molecule in order to get combined with starch grains. So what will happen? For that you first have to break cell membrane. Why do we break cell membrane? Because cell membrane is partially permeable or it is selectively permeable. It won't allow iodine molecule to seep inside the cell. Cell walls are fully permeable. Okay, so there's no need of breaking or damaging the cell wall. So we boil leaf in water so that we can break the cell membrane of cells. And another reason is to stop any chemical reaction which is going on inside the leaf. Chemical reaction means any metabolic reactions. We have studied in Unit 5 that enzymes are involved in all chemical reactions in living cells. Okay. So at very high temperature, when we are boiling water is having a temperature of 100 degrees Celsius. So these enzymes at 100 degrees Celsius will get denatured. Okay, so they will not get involved in any chemical reaction, and hence all chemical reactions they will stop, isn't it? So the first step is to take a fresh leaf from the plant and boil this leaf in water for 30 seconds to 1 minute. And the reason for boiling leaf in the water is to stop any chemical reaction which is going on inside the leaf cells and to break cell membrane. Okay, so that iodine solution can easily enter inside the cell and reach starch grain which is present inside the chloroplast of a cell. The second step is, after you boil leaf in the water, you have to take a test tube, okay, or you can take boiling tube. Fine. Now in boiling tube, place this leaf which is boiled inside the water, okay, and after placing leaf inside the test tube, cover this leaf with ethanol. You can take ethanol. Okay, ethanol is actually a type of laboratory alcohol. Okay, and after you cover leaf with ethanol in a test tube, place this test tube in a boiling water or hot water bath. Okay, you can simply say place this test tube in hot water bath for few minutes. Okay, now what happens after boiling it for a few minutes in the ethanol, you will see that this leaf actually releases. This leaf actually becomes pale yellow in color, or it actually the leaf gets decolorized and entire chlorophyll of the leaf gets released in the ethanol. Okay, this step is very important because if for example after first step, if we'll directly pour iodine solution on the leaf, what will happen? Iodine solution will be able to go and react to the starch grains, but the presence of chlorophyll in it, the green color of chlorophyll will mask the color of will mask the blue black color of starch and iodine solution. Okay, so it's important to remove chlorophyll out of leaf. Okay, and how to remove chlorophyll out of leaf? You have to take leaf, okay, put it in a boiling tube and then cover it with ethanol, and then you can place this boiling tube inside the hot water bath for few minutes. Okay, now one question which is asked here is, why don't we directly heat boiling, why can't we directly place this boiling tube on the flame of Bunsen burner or on the flame of the spirit lamp? This is because the ethanol's boiling point is actually around 70 to 80 degrees Celsius. Okay, so that's very less than water. And another reason is ethanol is highly flammable, so it might catch fire. And if for example, if it is not catching fire, then what can happen is it will get evaporate very soon. Okay, so that's the reason we keep this ethanol in the hot water bath. Okay, and another question which is asked is, what is the purpose of boiling leaf in ethanol? So the purpose of boiling leaf in ethanol is to remove all the chlorophyll from the leaf, okay, to decolorize the leaf. And the purpose of decolorizing leaf is, hence the color of iodine and starch, that is blue black color of iodine and starch, should not be masked by the green color of leaf. Okay, third step is, once the leaf is being boiled in this thing alcohol, the leaf becomes little brittle. Okay, the leaf becomes little brittle. Now when you'll take out this leaf and if you'll try to just open the leaf will break. Okay, because the leaf has now become brittle after boiling in the alcohol. So you can just dip it in the same hot water for 2-3 times to make it soft. So the moment you will you can just plug the leaf using forceps and just dip in the water and take it out. Dip in the water and take it out. Okay, so once you will dip this boiled leaf into the hot water for 2-3 times, the leaf will again become soft. Now what you can do is spread this leaf out on the white tile and drop iodine solution on it. Okay, now you are aware the color of iodine solution. Okay, the iodine solution's color is yellowish brown or brownish yellow, or you can just say brown or orangish brown. Okay, orange brown. Okay, so if after placing after dropping iodine solution on leaf, okay, and just wait for a few seconds and you can make out the appearance of blue black color. Okay, so for example, if you get blue black color, it shows the presence of starch. That means starch is present in the leaf. If you do not get blue black color and the color of leaf remains yellowish brown, okay, like as it is shown here in this picture. Okay, if the color of leaf remains yellowish brown or brownish yellow, that means starch is absent in the leaf. Okay, starch is not present. So remember, iodine solution on combining with starch gives blue black color. Blue black color shows the positive result or positive test for starch, and yellowish brown or brown color shows the negative test for starch. I'll just show you one image. Okay, this is the image of the real iodine test which is done on a leaf. Okay, so if starch is present in the leaf, you will get blue black color like this. Okay, and if for example starch is absent in the leaf, So you will get brown or yellowish brown color like this. And you can make out here the original color of iodine is yellowish brown. Fine. So you have to follow these steps in order to test the presence of starch in leaf. Okay. Now I'll just show you one small video. Have a look on that. So just have a look on this video. You will understand how to test the presence of starch in leaves. Okay. So you're aware that leaves of plants, they produce glucose through photosynthesis, right? And the excess of glucose present in the leaf is actually converted into starch. So you require green leaf, you require water, alcohol, that is iodine solution, boiling tube, spirit lamp, and the stand. Okay. So first what you have to do is you have to boil water and place a leaf in the water. Okay. You have to actually boil leaf inside the water. This will help in breaking down the cell membrane of leaf, leaf cells, so that iodine solution can easily seep inside the cells. Next, you have to take this boiled leaf inside this test tube and cover leaf with the alcohol. The alcohol used here is ethanol. Now you can place this test tube in hot water bath. After a few minutes, you can see that the entire chlorophyll of leaf has been released, or it is removed from the leaf, and it is released into the alcohol. Okay, this helps in decolorizing the leaf, so that the green color of chlorophyll should not mask the end result. It should not mask the color of iodine solution and starch. You can place this boiled leaf inside a petri dish, and now you can drop some of the iodine solution of the leaf on the leaf, and after a few seconds you can make out the leaf has turned blue-black in color. Okay, so the appearance of this blue-black color shows the presence of starch in leaf. These are the steps in order to test the presence of starch in leaf. I hope it is clearly understood, okay, with the help of this PowerPoint and the video which I have shown. In next video, we will discuss about how to test the importance of light for the process of photosynthesis. Take care till then. Bye-bye.